All right. Well, so thank you, Carlos. So now we're going to shift into a talk that's a little more focused on diagnostics and trying to understand AFM and from that approach. And so with that, I'll let Bernardo Mainou from the CDC get into his talk and tell us about those things. So go ahead and take it away, Bernardo.
Thanks, Matt. And hopefully everyone can see the right screen and
yeah, looks good.
And listen to me. This is one of the downsides of 3 monitors. So yeah, so this is going to be a slight shift in pace in the sense that I'm going to go fairly utilitarian and talk about our approach on really creating tools not only for clinicians but for us, for researchers and everyone else in between, to try to figure out what viruses are associated with AFM. I think we've heard a lot about this, and I won't really dive much on the background, and we definitely have focused on EVD68 on that end. So with that, I do want to start by thanking the team, because I have done 0 of the work that I will be presenting here. This is our current AFM team. Bryce Miller is starting to work on our ELISA development. Eric Roden has been central in creating an AVD68 microneutralization assay. Christina Roy has been working on a multiplex XMAP assay that I will talk quite a bit on today. And Nick Weiss has been helping us characterize some monoclonal antibodies that we have generated. So these are the people that really are driving the work that we are doing. And whenever and we've talked a little bit about this, but whenever we think about creating assays to figure out what is going on in the context of AFM, I tend to think of 2 bins. I think of, well, you can either detect the virus itself. And when I think about this, I think of 2 things. You either detect viral components like RNA or protein, a little bit like how Matt showed in those slides, where you can detect viral RNA in those neurons, or what we talk about viral antigens, so viral protein in the neurons. Or you can also isolate the virus, right? So you can isolate and culture the virus, and you can sequence it. And then the other end of this, and I would say Megan touched a little bit on this, is, well, you can look at what the virus infection has done in a patient and change things and see if you can find correlates of infection, in the sense of, okay, well, the virus was here. Can we figure out what it's done in the sense of host responses to virus infection? And Matt also talked about specific antibodies. But again, Megan touched about cytokines like interferons, and there's also changes in host gene expression. So you can use both of these sets to really figure out, okay, so what has happened and what is driving the infection, the disease that we're seeing. So currently, and again, Matt touched a little bit about this. When we get patients going into the clinic and we collect samples, and this goes from stool to CSF to nasopharyngeal swabs, and they come into us, our MO is we get these samples, and these are done through the CDC test directory. And they come into one of our labs where we look for the presence of RNA, right? So the actual virus, and then we can and this usually we can turn around and tell you, okay, there is signal for an enterovirus infection or a different, most picornaviruses. I think we can detect rhinoviruses to parecos to all sorts of other enteroviruses. And then depending on how things go, we can then take that and either isolate the virus and also sequence virus. So we have a group here that's generated and isolated a bunch of viruses that are now in VEI that we all use in the field. And then on our end, what we're really using is really focusing on serum and CSF. And this is using 3 types of assays to try to figure out if we can detect antibodies that are specific for, in this case, enterovirus D68, that can help us figure out if a kid has come in and has a respiratory infection, is this driven by a rhino? Is this driven by enterovirus D68? And these are the 3 assays that I will talk about. The microneutralization assay I'll touch on briefly, as well as our 2 assays that we use to detect binding antibodies, which are ELISA and this XMAP assay. I will say that all of these three assays are currently under development, and they're not CLIA approved assays and are not quite ready to go live. All right, so I touched a little bit on the microneutralization assay, and for those of you that have been doing polio work, you know that the neutralization antibodies are the gold standard for for polio, to detect immunity against polio. And Eric Roden has driven the work on creating a D68 neutralizing antibody assay. He has a beautiful assay which provides qualitative and quantitative data. This is an example from a publication from Eric, where he talks about the methods for this assay, showing 3 different enterovirus D68 antibodies where you can detect different levels. So here are the levels of antibodies. So basically the quantitative side of it, that you can detect antibodies from, basically the limit of detection on the bottom side is 2.5, and this is based on a log2 scale all the way to 10.5. So anything in between that's higher than 3 is seropositive, so going to the qualitative side. And anything below that, for example, in this sample, in this antibody, in this polyclonal antibody, human antibody, the enterovirus D70 antibodies were basically negative. So seronegative antisera, seropositive antisera with a level. One of the downsides to the assays, it does take 5 days because we're looking at cytotoxicity of cells. On the nice side, it's fairly good for on a high throughput side, where we can do about 200 to 250 samples per day. And we've been using this assay to perform sera surveys. Again, if you want to know more about this assay, I'll point you to Eric's paper that was recently published. So I want to talk a little bit more about 2 assays that we're developing that are more on the development side. And this is looking at really binding antibodies against EVD68. I'll talk a little bit about the ELISA initially, and actually I will say that before I go on to the actual ELISA, I will talk about a little bit about how the targets are using for the development of these 2 assays. And when we think of antibody targets especially for picornaviruses, but really enteroviruses, there's really two flavors. And when you think about the assay development, on the one side you can look at linear epitopes, so basically peptides. So short sequences that are either specific for enteroviruses or specific for some specific, for say, a specific family of the viruses like EVD68. Or you can think of looking at complex epitopes, like using the whole virus. You could also use other structures, but we've mostly focused on using whole viral particles. And really, when I talk about the ELISA, I'm focused on using whole viral particles to detect antibodies that are binding, and these are going to detect both linear and complex epitopes, as opposed to the multiplex assay that I'll describe later, which we're going to use linear epitopes to identify binding of antibodies to specific peptides. All right, so again, these are going to be our 2 assays. Our ELISA assay, if you're looking at what you're looking at, what the assay is, you have a piece of plastic, you bind, this is an indirect ELISA, we're binding the virus to the plate, and then we'll float, say, antibodies from either a monoclonal antibody or patient serum, and then we'll look at the binding through a secondary antibody that's going to emit light. Or in this case, light. And then the multiplex assay, which it's an XMAP or bead-based assay, which we coat these beads, and I'll talk a little bit more about this, with a peptide or a linear peptide. And then if the antibody binds and recognizes this amino acid sequence, the secondary antibody will then bind the primary antibody and emit a fluorescent signal. So this is really the fairly straightforward concept. And then I'll talk a little bit about the ELISA and some of the pros and cons. The ELISA assay was started and really developed by Kim Hetzler, who was a member of our lab until about 2-3 months ago. And Kim is now in Colorado and is now being taken over by Bryce Miller in her group. The ELISA will again detect levels of binding antibodies, and I want to distinguish the levels of binding antibodies versus neutralizing antibodies, because in the microneutralization assay, you really are focused on antibodies that are going to prevent the virus from infecting a cell, whereas here we don't actually know. This is just antibodies that can bind the virus. They may not neutralize or prevent infection. So it's a qualitative assay, so we can see if there's antibodies or not. And it's semi-quantitative, that we can provide a relative level on the levels of antibodies that are present in the sera. One of the nice things about the ELISA is that we can now just start getting into things like distinguishing antibody isotypes, whether it's IgM for a primary infection or IgG for someone that's been infected, sometime before. Or, there was a question earlier about IgA for mucosal antibodies, and we haven't really delved too much into IgA, maybe in the future once we have the IgM and IgG assays up and running. Even though, like I said, our focus is on the ELISA has been on the complex epitope side of things. So we're coating the plates with virus. And the assay is actually relatively short compared to the macronute assay. This assay only really takes 1 to 2 days, but the throughput is much less, right? It's about a log, I guess, if you want to think about it. So the macronute assay, you get 200 samples a day. The ELISA, you're only doing about 20 per day. So this is an example of the data of what it looks like. And this is using an ELISA against a clade B3 virus that's shown right here. And what you're looking at is the red lines show, this is for IgM on the left and IgG on the right. The red line shows the baseline, or basically what you would consider no signal. And all of the green dots that you see here are basically levels of signal. So if you have an antibody that binds really well to our virus, and then you probe for an anti-IgM secondary, you will get lots of signal. If not, you will go down here. And on IgG is the same thing, except looking at different antibodies. And one of the nice things about the assay is that you can start looking at basically teasing out what kinds of antibodies you may see in human sera. And these are different panel of human sera. And I'm going to highlight 2 sets. These 3 patient samples, for example, you can tell that they're positive for IgM but negative for IgG, suggesting that they had a relatively recent enterovirus D68 infection. Again, this is I have to be careful when I say we think they have antibodies that bind enterovirus D68, so they may have suggestive of an early enterovirus D68 infection. And then on the other hand, you see patients that have no IgM antibodies, but you see antibodies, IgG antibodies, like shown here and here, again suggesting that they were not infected with an enterovirus D68 relatively recent. And we're continuing to develop the ELISA and testing different matrices, going to expanding to CSF as well as different clades of enterovirus D68, so we can get this validated and hopefully get it out and more available for everyone. All right, for the rest of the rest of my time in the next 6-7 minutes, I'm going to talk a little bit more about this multiplex assay, which is I'm going to call it the XMAP assay. And basically the XMAP assay, it's been developed by 2 people in our group. Christina still started a lot of the development of this, and Chrissy has now moved on to another group here at CDC, and now it's been taken up by Christina Roy. When putting together this slide, I realized that one of the requirements to work on the development of this assay is that you must love the outdoors on your time when you're not at CDC. So similar to the ELISA assay, we're looking at binding antibodies, and it's still a qualitative and semi-quantitative assay that we can use to detect IgM and IgG antibodies. And we're going to focus in this case in the linear antibodies. We can use peptides to coat our beads. The assay is still relatively quick, and even though the throughput is pretty low, one of the huge perks of this assay is that you can look at multiple targets. So you can look at 50 plus different peptide sequences, for example. So how is that even possible? So the assay basically works by using magnetic beads shown here, which are coated with avidin, and then we can coat the beads with our peptide of choice shown here. Then you can float your patient's sera in here, and if you have binding to the peptide, then the secondary antibody recognizes it, and you get a fluorescent signal. And you can do this with the current instrument we have, about 50 different types of beads. So this would be one example where you have in one tube, you have 4 different beads with 4 different peptides, and you can measure different antibody bindings to the different peptides. All right, so what peptides are we using? And I'm talking, I think, I don't remember who talked about this. I think it was either Bill or Matt about the high conservation of I think it was Bill was talking about how the coronavirus capsids look very, very similar. And we've taken a little bit of advantage of this, and we have focus on creating peptides, or using peptides that are from the BP1 region of sterile enteroviruses. And for today, I'm going to focus on our scramble peptide as well as two polio peptides, and these are targeting the Poltave region, which are highlighted in red here, as well as the A71, a D70, a D60, and a D68 peptide. And I'm also highlighting in red how they all have this Poltave region that are shared in common, but especially this A71 peptide has very different amino acids outside of the Poltave region, which I'll come into play here in a little bit. And we also have an EBD68 immunoreactive peptide that we've identified that is unique to EBD68. It's in the C-terminal region of VP1. It's an outside of the Poltave, and we have found this peptide to be fairly intriguing. All right, so how does this look? So similar to the ELISA, what I'm showing here is on the y-axis. So anything up here is going to show the signal, right? So if you have antibody binding to our peptide region, you're going to get lots of fluorescent signal. And the three types of sera we're using to basically test the assay and validate the assay are a pan-polio negative human polyclonal sera. So hopefully pan-polio negative means that there is no poliovirus antibodies in there. And thankfully our two, this is just showing reactivity against our poliovirus 1 and poliovirus 3 peptides, and there is no signal. And then when we look in two sets of human sera, we see a nice dose-dependent response to it, meaning as you increase the levels of sera into the mix, you actually see more signal. So it's actually nice. So when there is no polio, there is no signal. When we have basically people you would have expected to be vaccinated against polio, you actually detect signal. All right, so it's either polio or I will say enterovirus antibodies that recognize the Poltave region. All right, so how do our EBD68 peptides perform in the same assay? And here I'm also including this extra control, which is it's human antisera that's been depleted for immunoglobulins, so there's really no signal here. And then when we look, and we've added now in this case an EBD68 specific peptide from the Poltave region, the same peptide that has basically the sequence in tandem 3 times, and an immunoreactive peptide sequence all the way at the end. Okay, so whenever we look at these, you get two fairly interesting patterns. You will see again, our polio antibodies are shown here, and then you see reactivity to the EBD68 peptide sequences in the one that's only 1, and then the 3 times over here. So basically, when you have the tandem sequence, you see a little bit more signal, and they all kind of cluster, suggesting that these antibodies are really recognizing that Poltave region. And the reason I say that is when we looked at this immunoreactive peptide that's very specific for EBD68 that's on the outside of the Poltave region, we really see no binding in our immune sera. And interestingly, in our pan-polio negative in sera, we actually see some signal. So what we're thinking, it's actually the assay is telling us, and the mixture of peptides are telling us, is that you're really seeing here binding to the Poltave region. So what we would characterize as enterovirus specific binding, whereas this EBD68 immunoreactive peptide actually shows a little bit more specific binding to EBD68. And then if we add 2 other peptides, this EBD70 peptide, it's also a very canonical Poltave peptide sequence. It basically behaves the same as our poliovirus and our D68 peptides. But when we add this EBA71, which has those sequences outside of the Poltave region that are very distinct, we see something slightly different, where this set of sera actually work, where you see all of the Poltave antibodies grouping together. In this second set of sera, they actually see some differences. And we're starting to try to use this peptides like this that have slightly different sequences to tease out some specificity to see if we can actually create a peptide sequence and an assay that will give you more than just an enterovirus binding and start giving you an idea of specific binding for a virus. We've kind of pushed a little bit some of our patient sera to see if we can really tease out whether this EBD68 immunoreactive peptide sequence can actually identify EBD68 specific antibodies. In this case, what we use is 3 different set of sera for 3 patients that had tested EBD68 negative by PCR, and then 3 patient sera that had shown to be EBD68 positive by PCR. On the right side, again, these are all of our Poltave binding or region antibodies, and they all have signal in the EVD68 negative patients. And then when we look at the EVD68 positive patients, the one really interesting thing we see is that we start really seeing, again, the positive antibodies are still binding, but we also only see this binding with this EVD68 immunoreactive peptide, again, suggesting that this peptide is really, really good at teasing out and finding EVD68 specific antibodies. All right, I'm going to talk one more little thing, and then I will finish up and hopefully leave some time, have left some time for questions. So this is one, I don't really have to talk much about this. This is an antibody that we got from Jim and from Jim Crow's lab at Vanderbilt. That's part of Matt's study, and Matt did not talk about this, but it's from the same cohort, and it's called 219. And we have been using this antibody to characterize our validate our assay. One of the really interesting things about this antibody that we have found is that it beautifully binds this immunoreactive peptide. And here's the immunoreactive peptide sequence. And this is an antibody dilution showing that little antibody, you don't see a lot of binding. And as you add more antibody to the mixture, you actually see beautiful binding. So we're really excited because this antibody is going to be a beautiful control as we move forward with our assay to validate it, but also shows that you can actually use this assay to identify binding regions for antibodies. And now we're also hoping to play around with this peptide sequence and see if we can lose the signal to this antibody. And we're working with Matt on this side. And just for the sake of time, I'm going to leave the conclusions here from what I've hopefully shown you, and then we'll move on to questions. And I'd like to also thank the people in blue that have contributed a lot on a lot of the work that I showed today, as well as the rest of the group here at CDC from our branch and our laboratory, as well as alumni that have worked hard on EVD68 stories. Hopefully I left a little bit of time for questions.
You did indeed, Bernardo. Thank you for that. And we have a good question in the chat, but I'm going to take privilege of being the host and ask, so for that last set of data you showed where there were 3 patients and only 2 of them, even though we knew they were PCR positive, only 2 of them had reactivity to that specific peptide. Do you know how recently after that positivity that sample was shown? So it's like, did you just get the blood too early or the serum too early and maybe they would have made that? Or does it show that your peptide maybe isn't going to catch everybody? Yeah.
Yeah, I think it's a mixture of both. So and the answer is complicated. I need to go back and look at the data set for that. So for some of the antibodies in our cohort, we do have the time of collection, and for some of them we don't. So I need to double check on how close to when they tested positive for D68 was the sera collected. And for some antibodies that we have parent samples that, for example, a stool was collected that tested positive by PCR, and then a subsequent serum was collected. And sometimes we have that information and sometimes we don't. But then going back to the other part of the question is, it's not going to be perfect.
Yeah.
So I think, biological samples, you're going to miss some things. But I've been very surprised that the one peptide sequence has been relatively good at distinguishing between EVD68 samples and other enterovirus samples, just because of, again, what Bill touched on and what we all know, which is enteroviruses share a lot of a similarity, and especially for binding, even with polio. I mean, you have polyantibodies will bind between all serotypes, but it's a neutralization that it's really where the beautiful part comes in as far as immunity.
All right, well, I think that dovetails nicely into this question here from an anonymous attendee. All right, so how do we know that the neutralization titers to enterovirus D68 are generated by exposure to enterovirus D68 versus representing heterotypic antibodies in response to other non-polio enteroviruses with cross-reactive neutralization? And can your assays be used in a serosurveillance, seroepidemiology way to understand, this question, I guess, about cross-reactivity or not, which I think, sounds like you're getting into that, but.
Yeah. I mean, I think that's, and that's something that it's been talked a lot about, especially from by Amy Rosenfeld and others. Yeah. Yes. So I will say I think for polio, we know a lot more about the neutralization what the neutralization titer data means, right? Specifically within specific polio serotypes. For the jury's still out for enterovirus D68 on whether you can actually say that the neutralization, that if you have an antibody titer by a neutralization antibody titer for one, say clade B1, whether that means that that person had been infected by B1 or whether it's cross-reactivity. What I'm offering is a tool to answer those questions, and actually I'm offering several tools to answer those questions. I just think the jury's still out, just like I still feel like the jury is out on whether and we'll hear more about this on whether a vaccine or antibodies to what level they can protect from paralysis. Yes, all I'm saying is that the assays will tell you if there's antibodies. I think we all need to get more data on the levels of protection and what does that really mean.
Yeah, awesome. Well, can I have you unshare so we can move on? But no, I think, Bernardo, this is really important to answer that question. And in the end, as this is a perfect segue, like you said, into the next talk. It may not matter where the antibodies came from, as long as you have the damn things and they can protect you. That's what we care about, right? But it's certainly, from a scientist's perspective, very important that we try to understand where they came from. So